Hello students, today we are going to study an advanced form of microscopy, that is electron microscopy, and we will see how electron microscopy is better than light microscopy. First, why electron microscopy? So, the very best light microscope, it has a resolution limit of approximately 0.2 m, but because bacteria usually are around 1 meter in diameter, so only the general shape and major morphological features are visible with the help of light microscopy. To increase the resolution of a light microscope, we have to decrease the wavelength, and this is how electron microscopy is better than light microscopy, because the electrons have the wavelength around 0.005 nanometers, which is approximately 1 lakh times shorter than that of the visible light, and thus electron microscopes have a good resolution as compared to the light microscopes. Transmission electron microscope has a better practical resolution, which is roughly 1000 times better than the light microscope. This is a figure, a comparative figure of light microscope and transmission electron microscope. In case of transmission electron microscope, the source of electron is this tungsten filament, and this is actually a part of the electron gun. As you can see in this figure, the heated tungsten filament in the electron gun, it generates a beam of electron that is focused with the help of the condensers onto the specimen. So these we have condenser magnetic lenses. Why magnetic lenses? Because since we have electrons and the electrons cannot pass through a glass, so we have condenser lens made up of magnets to focus them onto the specimen. One more thing of importance here is that this transmission electron microscope, this is actually in vacuum. Why in vacuum? Because electrons will be deflected by the air molecules. So electron microscopy is done in under vacuum conditions. So they are operated under high vacuum in order to obtain a very clear image, since electrons are deflected by the collision with the air molecules. Now, when the beam of electrons strikes the specimen scatters the electrons passing through it, and now the beam, after passing through the specimen, is focused by the help of magnetic lenses to form an enlarged visible image of the specimen on the fluorescent screen. Since the specimen has both denser and lighter regions, when the beam of electrons strikes the denser region, the specimen scatters more electrons, and therefore those regions appear dark in the image, since only few electrons strikes the area of the screen. Whereas the lighter regions, or we can say the electron transparent regions, they look brighter, and this is how we can see darker, denser regions and the lighter regions in the specimen. This is the comparative study, since you can see the resolution, the magnification, and how light microscope differs from the electron microscope. The light microscope has just a magnification of 1000 to 1500, whereas electron microscope has 1 lakh. The best resolution in case of light microscope is 0.2 mu m, and whereas in case of electron microscope it is 0.5 nanometers. Radiation source here is visible light. We have electron beam in case of electron microscope. Medium, in light microscope we have air. In electron microscope we have high vacuum. Here you can use glass lenses, but in case of electron microscope we have electromagnets. The contrast is because of the differential light absorption in case of light microscopy, whereas in case of electron microscopy we have electromagnets. In case of electromagnets, and it's basically because of the scattering of the electrons that creates a contrast. The focusing mechanism, we have lenses. Here we have magnetic lenses. Again, for change of magnification, we have in light microscopes we have objective lenses or the eyepiece, whereas in case of electron microscope we have the magnetic lenses. We adjust the current to the magnetic lenses, which actually changes the focusing and the magnification power of a microscope. Here, in case of light microscope, the specimen is mounted on a glass slide, whereas in case of electron microscope, the specimen is mounted on a metal grid. Next, we have the scanning electron microscope. In this type of microscope, the only difference with the TEM microscope is that here the secondary electrons forms the image. This is the figure, diagrammatic representation of how the scanning electron microscope work. We have electron gun, we have condenser lenses to focus the primary electrons onto the specimen. But here the specimen actually generates secondary electrons which reaches to the detector, and the photomultiplier converts the light signals into the electric signals which are recorded on this cathode ray tube. So in scanning electron microscope, the secondary electrons actually forms the image, whereas in case of transmission electron microscope, only the primary electrons forms the image. So this was all about secondary electron microscopes, scanning electron microscopy and transmission electron microscopy. Thank you.